Hi everyone! In today's video, we're going to talk about bacterial growth. We're going to divide this up over two videos to keep them nice and short. Bacterial growth really means two things. There's the division of individual cells, where one cell becomes two, which is what this video is going to cover. And then there's growth of the entire population of bacterial cells, where we start with maybe 10 cells, and after multiple generations of growth, those become 100 million cells, or however many you have in a culture. So first, we're just going to look at the division of individual cells. Bacteria divide by a process called binary fission. Binary fission is much simpler than mitosis, because if you remember, bacteria only have one chromosome. And a lot of eukaryotic cell division is based on lining up and separating the sister chromatids, but that's not a problem for bacteria. The first step, much like with mitosis, is to replicate the DNA. We need to have two copies of that chromosome in order to divide. So up here, you can see our bacteria has already replicated its chromosome, and we now have two copies. You can also see that these chromosomes have been attached to the cell wall. This is what's going to allow them to separate out between the two daughter cells. So no spindle fibers, just attach them to the cell wall. Nice and easy. The second step, the easiest step really, is that the cell gets bigger. Our bacteria cell grows from the middle outward so that it elongates. As it elongates, the chromosomes are separated out. The third step, where things start getting a little more complicated, is the formation of what's called a cross wall. It can also be called a septum or a divisome, but cross wall is the easiest word. So that cross wall forms, and it really begins to pinch inward. And this pinching is what's going to allow for separation. The fourth step, we're just continuing forming that cross wall. You can see that it's going to be completely formed right here. And the two cells are still stuck together at this point. And then the final step, completely separate those cells. So complete division. And now we have two daughter cells. Pretty simple, right? Especially compared to eukaryotic cell division. So that's what each cell does when it replicates and it divides and becomes a new cell. The process can take varying amounts of time, but as you can imagine, because there are less steps, it's much easier than mitosis. There are a few proteins that are really important in the process of cell division that I would like you to know about. The first is called Fitzy. Now this protein is usually found in monomers diffuse throughout the cytoplasm of our bacterial cell. However, during the process of cell division, it will polymerize into long filaments. These filaments are very important at the final stage of cell division because they are actually what separates our two daughter cells apart from each other. The localization of Fitzy during the process of binary fission can be seen in the panel that I've just numbered. Here in panel 1, you can see that it is diffuse because this cell is not yet dividing. As time progresses, however, that Fitzy begins to centralize or localize there in the middle of the bacterial cell. And all the way in panel 4, if I outline our two cells that are trying to divide, you can see that Fitzy is almost entirely there in the middle where the septum or the divisome would be found. These filaments of Fitzy are called the Z-ring, and they contract to pinch apart the daughter cells. So imagine what a cell would look like if it tried to divide and was deficient in Fitzy. Say Fitzy couldn't polymerize or it couldn't carry out this pinching motion. What do you think the cell would look like after it had completed all of the other steps of binary fission? Next up in our important proteins for cell division is PARM. Like Fitzy, it's found as monomers, diffuse in the cell, and during cell division it polymerizes to carry out its function. And its function is to separate the plasmids that a bacteria may have. So if we just draw a little bacteria here. They have their chromosomes, and as we saw in our last picture, those chromosomes will attach to the cell wall, and that's how they'll be separated out during division. But plasmids don't get attached to the cell wall, and we want to make sure that those plasmids get equally distributed between daughter cells as well. So PARM works in conjunction with a protein called PAR, and PAR actually binds to the plasmids. We can see that right up here. So this little blue squares, that is PAR bound to the plasmid. PARM then polymerizes. The polymerization of PARM pushes our plasmids apart from each other as the cell elongates. So it pushes plasmids as the cell grows, as the cell elongates. Until finally, in our last step, these cells have completed division, and now PARM is going to depolymerize after division is done. So we've separated out our plasmids, we've made our 2 new daughter cells. Both daughter cells have everything they need to continue growing in their particular environment. So there's one more protein that I really want you to know about, and this one is called MREB. The function of MREB is to provide shape to elongated cells, so to our bacilli. So like with Fitzy, like with PARM, it starts out as free monomers in the cytoplasm, and it's going to polymerize to form chains. That's shown pretty well in our figure here, where we have on the left side of our cell, we have all these individual MREB proteins that are free in the cytoplasm. They polymerize to form these sort of ribbon-like helices. They go throughout the cell, attaching next to the plasmid membrane, and they would go from, in a fully polymerized cell, they would go from one end all the way to the other. We can draw that pretty poorly if we just do this. So from one end all the way to the other. Now think about this. If a cell lacked Fitzy, what would be the result? What would a cell look like if it lacked MREB? What kind of structure would it have? And what would happen if a cell had deficient or mutated PARM? Okay, so that's the end of part 1. We've covered the specifics of binary fission. We've talked about Fitzy and the Z-ring and its role in that final stage of separating our 2 daughter cells. We've discussed PARM and how it's useful and necessary for separating plasmids out between daughter cells. And we've talked about the importance of MREB in providing shape to our elongated bacterial cells.